All right, everyone, please be seated. Good morning, ladies and gentlemen of the jury. Good morning. Hope you had a good evening. Appreciate you being here on time this morning. And I do want to ask, I trust that everyone was able to abide by the court's instructions and avoid any exposure to media coverage. Any problems? No. Okay, all right. Thank you very much, and we'll get right to it. If the state would please call the next witness.
Your Honor, at this time, the state calls Michelle Shephard to the stand.
Good morning.
Good morning.
If you would, please raise your right hand.
Do you swear and affirm the testimony that you're about to give will be the truth, the whole truth, and nothing but the truth, so help you God? I do.
Please be seated.
May I inquire, Your Honor? Good morning, ma'am.
Good morning.
I'm going to ask you just to try and stay close to the microphone. This courtroom has a tendency to eat up the sound. Can you please tell the court reporter your full name and how you spell it?
Sure. My name is Michelle Shephard. Michelle is spelled M-I-C-H-E-L-L-E. Shepherd is S-H-E-P-H-E-R-D.
Can you tell the jury where you were working in December of 2022?
Yes, at the Georgia Bureau of Investigation, the Division of Forensic Sciences, otherwise known as the State Crime Lab, in the Coastal Lab in Pooler.
So you worked at the Coastal Lab?
I did.
Okay. And at that laboratory, what was your title and what were your responsibilities?
I am and was a forensic DNA analyst and biologist.
And as a forensic DNA analyst and biologist, how long were you employed in that capacity as of December 22?
22 years.
Had you always had that same title?
Various titles with the state, but in the forensic biology section.
Okay. At some point, did you leave that position?
I did.
Why did you leave?
I left after 13 years at the headquarters lab up in Atlanta. I left to go to the private sector, still working in human identity to manage a forensic tech support and training team with a private vendor.
And so in total, how many years did you serve as a forensic biologist?
38.
And are you currently working in that capacity?
I retired a year ago.
Okay. Can you tell us what educational background you were required to achieve to be able to perform that position?
Sure. I received my Bachelor of Science in Laboratory Technology from Auburn University. And while I was in school, I actually worked with the Alabama Crime Lab system. I received training upon my graduation once I started at the headquarters lab with GBI and received advanced DNA analysis methods training at the FBI Academy in Quantico. I do have graduate coursework through the University of Virginia and the University of Alabama at Birmingham.
Were you required when you became a member of the GBI to do any initial training prior to taking on your own individual casework?
Yes, ma'am.
And how long was that process?
Over a year and a half, probably.
We've heard testimony already about what peer review is.
Yes.
Is your work in the biology section also peer reviewed?
Yes, ma'am. In the forensic biology section, 100% of our results are technically and administratively peer reviewed.
And does that mean somebody analyzes the work you did to ensure that it's accurate?
Yes, if I can elaborate. It means that another qualified DNA analyst reviews all of my notes, all of my controls and standards and results, and ensures that they would come to the exact same conclusions that I did.
Can you describe for the jury what the responsibilities are of a forensic biologist or DNA analyst?
Certainly. We receive evidence from peace officers submitted from across the state of Georgia to generate or develop DNA profiles from that evidence from body fluids and stains of body fluids.
What types of body fluids do you examine in your position?
Primarily blood, semen, and saliva.
And are you able to also analyze substances other than just fluids?
Yes.
What else do you analyze?
Tissues, bones, organs.
Is that anything that potentially might have DNA?
Correct.
Okay. And sometimes, just to be clear, is DNA always in, meaning human form, meaning, I guess I'm trying to be clear on how I phrase this. You've mentioned a lot of things that are human body, organs, or tissue. Can you retrieve DNA from other substances that are not necessarily human?
Yes. All living things have DNA, so DNA stands for deoxyribonucleic acid. It's a chemical that's found in virtually every cell in the human body and in all living things.
And the crime laboratory, do they only handle human organisms?
Sometimes they're mixed with bacteria, which are living things, and other organisms. But the testing that we do is directed and specific for human and higher primates.
And are you trained in the handling and preservation of those biological samples?
Yes, ma'am.
Can you tell the jury what that process involves?
So DNA is a protein, so it is susceptible to the environment, UV light, sunlight, heat, humidity. Those things are detrimental to DNA and can start to break it down. So we always want to preserve the evidence that may contain DNA in a manner that will preserve whatever is already deposited.
And are you able to ensure that protocol is in place at the crime laboratory as well?
Yes.
And can you tell the jury how you do that within the crime laboratory?
So within the lab, once an item of evidence is submitted by a peace officer, our evidence tech staff will take that item out of a lockbox. It's like a locker that the officer deposits the evidence in sealed packaging, and then our evidence retrieval on the opposite side. They will open the package, describe it, and apply a unique crime lab case number, item number, and description into the computer, so that as that item of evidence moves throughout the lab, what we call a chain of custody is maintained, so that it's tracked and documented who had access to that item of evidence as it moves through the lab.
And in order to do that, do you oftentimes affix any information on a sample?
Yes, or the bag, the packaging containing that sample, there'll be a barcode, And as we transfer that evidence, we ourselves have identification tags that we scan that barcode and enter a PIN number, just like you would at an ATM, to track what individual had access to that item, or if we're placing it in a vault or an evidence locker, those have barcodes as well.
Now, can you explain to the jury a little bit what DNA actually is?
Sure. So, as I said, it stands for deoxyribonucleic acid. It's a chemical that's in almost every cell in our bodies. There's really about 4 things for you to really recall or know about DNA. The first is that we are all unique. So at the union of sperm and egg, we get half our DNA from our father and half from our mother. So when those combine to create a new human being, that person's DNA is set at conception and remains the same throughout their life and even past death. So that's very important to know, that unless you have an identical sibling, there's no one that's going to have the same DNA as you. Secondly, the DNA molecule, although microscopic, is long, and the majority of that DNA is the same from human being to human being. And that makes sense if you think about all of the multitude of enzymes and proteins that the DNA codes for in our bodies. But there's also a portion of the DNA that is very different from person to person. And that's the areas of the DNA that forensic scientists study or interrogate. Thirdly, if you take a cell from my foot or my liver or a buccal swab, the lining of my cheek, all of that DNA from one person's body is the same. So that allows us to compare, say, sperm cells to blood cells to saliva. And those are important things to remember in a forensic context about DNA. And
so you indicated those specific unique areas are what you focus on, is that right?
Yes.
Okay. How do you determine whether a biological sample is suitable for DNA analysis?
So a portion of our protocols or procedures are to, once we break open the cells and get to the DNA, if you will, we perform a step called quantitation, and that is where we're quantifying or finding out about how much DNA is present in a sample and the quality or how suitable that DNA is to move forward in the procedures.
Is every biological sample then eligible for DNA analysis?
Not always. Sometimes samples will be too degraded, which means the protein DNA has been broken down or degraded to the point that it's not suitable to give us results.
What about quantity? Does that bar you from being able to do DNA analysis at sometimes?
Sometimes it does. If we don't have enough, or if the pieces, if you will, of DNA are not large enough, we are unable to get a result. One of the beauties of DNA is it doesn't morph or change into something else. It fades so that we get no result instead of we don't get a different result, we just get no result.
And does DNA remain after death?
Yes.
Does the GBI Coastal Crime Laboratory have accreditation for DNA analysis?
Yes.
And was that current at the time that you were working there on this case?
Yes, ma'am.
Can you explain to the jury what type of DNA analysis you perform at the GBI Coastal Crime Laboratory?
So one of the services we perform is DNA typing, where we have an unknown from a crime scene, say a car has been broken into and there's blood there. The officer will submit a swabbing of that sample, and we develop a profile from that using a very stepwise procedure called a protocol, very similar to if you're cooking in a kitchen, you're going to follow a recipe. So we follow those stepwise steps. The first is extraction, where we're going to take a small cutting of that swab or that tissue from a crime scene and place it in a test tube. And we add chemicals and detergents that break open the cells and remove the other proteins that are not useful to our studies. The next step is the quantitation that I talked about, where we're determining how much DNA is there and if it's suitable for additional testing. The next step is called amplification, and this is where we make millions, if not billions, of copies of the regions of the DNA that we want to study. And we do that using different instruments in the laboratory. Then the next step is to analyze or separate all of those pieces of DNA, and we use an instrument that produces a graph, a printout with a series of peaks and valleys that has letters and numbers associated with those, and that is what we refer to as a DNA profile. And that profile is unique to each individual. So we've done that for the unknown, and then separate in time and space, we perform those same steps with a known, a sample that is collected through venipuncture, so a blood sample, or from autopsy, or from the lining of someone's cheek. That is considered a known because we know who that came from. And then we compare those two profiles to see if they're the same or different. If they are different, that's called an exclusion, and that means that that known person could not have contributed that unknown sample from the car, in my example. But if we compare them and they are the same, that's called an inclusion or a match. People refer to it as a match. And then we use statistics to develop a frequency to relay how common or rare that match would be in the community.
And so are those the steps that were taken in connection with this case?
Yes.
Okay. Approximately how many forensic cases, I said approximately, have you worked in your career?
Tens of thousands.
And in each case, is there more than one sample?
Usually, yes.
So that tens of thousands is multiplied by however many samples were in each case?
Correct.
Now, do you only do DNA analysis for criminal investigations?
Yes, with the state of Georgia.
Okay.
Yes.
And when I say criminal investigations, do you handle suicides? Meaning if there was a suicide, might you get a sample from a suicide case?
Yes.
And that
yes.
might not have been criminal in the nature of simply somebody killed them, they took their own life, correct?
Yes, ma'am.
Okay.
I misunderstood.
So, no, that's okay. What about missing person cases? Do you
Yes.
sometimes do that as well?
Yes.
Okay. So it's oftentimes law enforcement that's making the submission, but you don't always yield a criminal suspect in a case, do you?
Correct.
Sometimes you said there are exclusions, right?
Yes.
So you would potentially exonerate someone even.
Well, I would exclude them, and then the court system would.
Correct. My
exonerate.
apologies. I don't
That's
mean to
fine.
put farther along, but you would indicate that that was not them.
Correct.
Okay. Now, have you ever worked on cases where DNA was not suitable?
Yes.
And how do you document that for the purposes of your records?
So for every test that we perform in the lab, we produce a written record or report to submit to the law enforcement agency that presented the evidence, as well as to the courts. So that report would read something like there was insufficient or inadequate DNA, so not enough DNA for further testing.
And would you then stop at that step?
Yes.
Okay. And if there is sufficient DNA to do additional testing, is that the process that you just described for the jury?
Correct.
Okay. Have you prepared reports on each of the cases that you just described in the tens of thousands that you previously worked?
Yes.
Is a report required in every case?
Yes.
And is that report also peer-reviewed?
It is.
Have you ever been asked to review or validate the work of other forensic biologists?
Yes, I am.
And would that have been done during the entire time that you were at the crime laboratory as well?
It would have, yes.
Have you previously been qualified as an expert in forensic biology in the courts of this state or any other jurisdiction?
Yes.
Can you tell us approximately how many times?
Over 300.
And in that capacity, was that in the field of forensic biology?
And DNA analysis.
And
yes.
DNA analysis. Have you ever provided testimony, or rather been asked to provide testimony where you were denied expert status?
No, ma'am.
And can you explain to the jury where you've testified? What parts of the state?
All over the state of Georgia, in federal courts in Wisconsin, civil courts in Wisconsin and California. Yes.
So all over the country, not just Georgia?
Yes.
Okay. At this time, Your Honor, the state would like to tender Michelle Shephard as an expert in the field of forensic biology and DNA analysis.
Any questions from the defense?
No questions and no objection.
All right, then she is qualified in the field of forensic biology. You may proceed.
Thank you, Your Honor. Ms. Shephard, were you provided with biological evidence to analyze in connection with the case that we are here for today?
Yes, ma'am.
And did you, in fact, perform the analysis on those substances?
I did.
And ultimately, did you generate a report in connection with this case?
Yes, I did.
In fact, did you do more than one
Yeah,
report?
I did, yes.
Okay. I'm going to ask that I show the witness 1.69 and 1.70. I'm showing them to counsel. They've been pre-marked, Your Honor. Thank you. May I approach?
Yes, ma'am.
I'm showing you these two reports. If you could just take a look at those.
Yes, ma'am.
Do you recognize those reports?
I did.
And how do you recognize them?
I recognize states 1.69 and 1.70 as the reports of my testing at the crime laboratory for the case with the victim as UID Liberty, which is unidentified from Liberty County, case number 2022-600-7185.
And is your signature affixed to those reports?
It is
Is that your actual signature?
an electronic signature. Yes.
But it's the one that you placed on it electronically?
Yes.
Are those exact photocopies of the reports that you generated in connection with this case?
Yes, ma'am.
At this time, Your Honor, I seek to tender 1.69 and 1.70.
Any objection?
No objection, Your Honor.
Thank you. I'm now approaching, Your Honor, with that was 1.69 and 1.70, correct?
Correct.
May I approach, Your Honor, with these additional exhibits? I'm handing you what has already been introduced as exhibits 1.63, 4, 5, 6, and 7. And I'm going to ask you if you recognize what those are.
Yes, ma'am.
And what are those?
So I recognize State's Exhibit 1.63 through 1.67 as sealed envelopes that we received from autopsy with various bloodstain cards from partial human remains.
And when you say you received them from autopsy, would that have been from a medical examiner?
Yes.
Okay. And do you have any initials or signatures on any of those exhibits, I believe it's 6.3 through 6.7?
I do. In each of these envelopes, which is sealed, there is a unique crime lab case number, item number, barcode, my initials, and the date that I analyzed these, as well as on the seals, have my initials and the date in my handwriting.
So if you were to receive an item in a closed fashion, meaning sealed,
Correct.
how do you make sure that it gets not tampered with?
So the rule is you always, you don't want to, if it's possible, open on someone else's seal. So you create a new opening with scissors or a razor. Again, this is in the laboratory with all personal protective equipment on in a sterile environment. And then once I analyze it, I affix the evidence tape and seal it with my initials and the date as it's to be passed to an evidence locker or another individual for further testing.
And is that done on every sample?
Yes.
If I may approach, Your Honor. I'm going to trade you.
Okay.
I'll have those back. I'm going to publish, Your Honor, if that's okay.
You may.
1.63. My apologies.
There is a short delay in that.
It's okay. I'm pointing to the lower right-hand portion where there is a barcode. Is that correct?
Yes.
What are those numbers right there?
So the 2022 dash is the crime lab case number that was assigned on entry of this evidence. So each item in this case will have that unique identifier, and then below it is, it says SUBM, that is submission number 5 in this case.
And so if I scroll in close, and I turn this on its side so it's easier,
Okay.
what is right here? Do you see that?
I'm sorry, I didn't see what you're pointing to.
Right here, along the tape.
Oh, the red evidence tape on the short side of the envelope, and written on the envelope as well as the tape is MS that's my initials, and the date, which is 12/13/22.
And so would it be fair to say that on each of these exhibits, that same procedure, we're just placing them on top of each other. I'm now showing 1.64, 1.67, 1.66, and 1.65. That same procedure was taken with respect to all of those cards?
Yes, ma'am.
And when you received them, were they in a sealed condition?
Yes.
And what did you do with these cards when you received them?
So the service that was requested was to develop DNA profiles from each of these bloodstain cards to see if they originated from the same individual.
So at the time you received them, was there any marking on the card to indicate for you what portion of the remains it was from?
Yes, ma'am.
I'm going to show it again. Or I'm just trying to make this quicker by Elmo. I don't know if that's the technological word for that, but the overhead presenter.
Okay.
And I'm going to scroll in here, and on the lower left corner, is there a label affixed to 1.65?
Yes, ma'am.
And-
It
ahead.
says UID Liberty WF, which is unidentified, remains from Liberty County, W for white, F for female, and then right leg.
I'm now showing 1.66.
This one is from left leg.
1.67,
It's labeled head.
and 1.64,
Torso, spleen,
and 1.63.
torso.
So were there in total then 5 separate bloodstain cards that you received?
Yes.
And did you create a profile for each of these 5 portions of the remains?
I developed a profile using the procedure that I described earlier for each of these samples.
Without sounding graphic, you did not receive her remains, correct?
Correct.
Just those cards?
That is correct.
Okay. And can you tell the jury, did you follow that same procedure and protocol for each and every one of those 5 samples?
I did.
How did you do that with a bloodstain card?
So the bloodstain card contains a piece of material, kind of like a piece of gauze or a piece of paper. And so I took with sterile scissors a small cutting of that and put it in a tube and subjected it to our procedures.
Okay. And were you able to obtain a profile on each of those 5 bloodstain cards?
I was, yes.
And what were the results and findings of your analysis?
So DNA was isolated from each of the 5 cards we just presented. And the DNA obtained from each item contains a single profile of the same individual.
So at that time, although she was still unidentified white female, would it be fair to say that we knew then that she was at least all the same person?
That is correct.
Okay. And did you document that in your report?
I did.
If I can approach Your Honor. Can I have 1.69, please?
You may.
Thank you. May I publish, Your Honor? So in that very first paragraph there's a whole lot of letters and numbers.
Yes.
Can you explain to the jury what a loci is?
Yes, so a loci is a locus, is single, loci is plural. It's a location think of it as a street address for DNA. So it tells a DNA analyst where on the molecule exactly, very specifically, we are getting results.
And so were those the loci that you were looking for to determine those unique characteristics for each individual person?
Yes.
And now you mentioned some statistical data. Can you tell us what the frequency was in this case?
Yes, so the fourth paragraph reads, the frequency in which the profile obtained from these 5 bloodstain cards would be expected to occur in a given population in approximately 1 in 300 septillion.
Do you know how many zeros is a septillion?
There are 24 zeros in septillion.
So 300 would be the number 3 with 26 zeros after it?
Correct.
That's the likelihood of it being the same person, correct?
That is an approximation of how many other individual DNA profiles I would have to look at before I would expect to find another just like this.
That's 26, right? Zeros?
Correct.
And after you made that first analysis, did you then subsequently make the report we just looked at?
Yes.
And can you tell the jury, did you know who that person was by the profile?
No.
Why not?
In forensics, we have to have a known to compare it to see if they're the same or different. And at that point in time, there was no known reference sample to compare it to.
Would it be fair to say, again, I'll just show it to you, that this report, I'm showing it to the witness, Your Honor, was generated back on December, up here,
Yes, 16.
December 16th. So in December 16th of 2022, you didn't have a known sample to compare it to yet, did you?
Correct.
Okay. Can you tell the jury what CODIS is? C-O-D-I-S.
Yes, so CODIS is an acronym that stands for the Combined DNA Indexing System. It is a series of computers and software curated and managed by the FBI that allows forensics and law enforcement to compare samples to individuals and individuals to, it allows us to compare evidence profiles to evidence profiles, as well as evidence profiles to individuals.
Is there a very specific protocol for what is allowed to be in the CODIS system?
Yes, ma'am.
Okay. So only if those are in compliance would it be within CODIS?
Correct.
Now, does that allow you an opportunity to attempt to try to identify an unknown sample, even if you don't have a known sample?
Yes.
Doesn't always work, though, does it?
Correct.
Okay. So it's just one step in the process that is an option to try.
Yes.
Now, can you tell the jury what a buccal swab is?
So a buccal swab or a buccal swab, depending on different people say it different ways, it's spelled B-U-C-C-A-L, and that is a cell that lines different orifices in our bodies. People usually refer to a buccal swab as taken from the inside lining of your cheek.
And can you tell the jury what is the difference between genetic genealogy and DNA?
So DNA testing and typing is done in the forensic laboratories across the world to compare knowns to unknowns to see if they have the same individual contributing that unknown sample. Genetic genealogy is another branch of forensics that seeks to use still DNA technology, but to build family trees to see, since I've testified to the fact that we get half our DNA from our father and half from our mother, we share DNA with our parents and also with closely related individuals. And genetic genealogy studies and expounds that to try to develop leads and generate closely related individuals.
So when you actually have a DNA sample, in this case you were able to actually analyze the material beyond just potentially genetic genealogy. Is that correct? I mean, maybe that's the wrong way to phrase it. Can you just describe then the difference between the two of them, what they can do? Does that make sense, what I'm asking? If I need to rephrase it, I can. I'll withdraw the question. What I'm asking you essentially is DNA analysis of an actual sample able to give you a definitive result? Meaning, in this case, you produced an actual profile, correct?
Yes.
And you were able to determine whether or not in that frequency those each, and I will use the word match, but to that frequency were the same sample profiles.
Correct.
And you indicated that they were.
Yes.
Okay. Now, with respect to the buccal swab, I believe you called it a buccal swab, that was used, did you have a buccal swab in this case at some point?
Yes, I did.
And do you have that up in front of you now?
I do.
Can you tell us what exhibit number that is?
So I identify states 1.71 as my crime lab case number associated with this case. It is item number 61, and it is labeled buccal swabs collected from Betsy Mebane.
Okay. And can you tell us, how does one collect a buccal swab?
So a medical practitioner or law enforcement agent will take a cotton-tipped applicator, it's like a big Q-tip, and just simply swab the inside of your, your cheek, and that removes the cells of your cheek, and then places that in a container to air dry, and then places it in an envelope.
And do you recognize that to be the buccal swab that you received in this case?
Yes, ma'am.
Your Honor, at this time I would seek to tender that exhibit before the court. Thank you. May I have that for just a moment?
Yes.
May I publish, Your Honor? I'm going to just show it to the jury. And does this swab ultimately have the exact same procedure and protocol with the barcode and your initials that you just previously described?
Yes, it does.
And I am not going to open this here.
That's best.
This contains biological evidence, correct?
Yes.
Okay. So you would have opened this in a sterilized environment?
Correct, with gloves and a mask and face shield and lab coat, etc.
Is that how you handle every sample?
Yes.
Okay. So this sample, what did you do with it?
So that is considered a known. So on receipt of that, I submitted it to the same procedures that I've described and made a comparison of the profile that was generated from that sample to the previous 5 envelopes.
And were you able to generate sufficient DNA to create a sample from that buccal swab?
I was, yes.
And did you in fact compare it to the 5 portions that you just described previously?
I did.
And what were your findings as a result?
That my conclusions are that Betsy Mebane is not excluded as the biological parent or biological relative of the bloodstain cards, the 5 bloodstain cards, with a probability of maternity of 99.9999%.
Can you tell us why it isn't 100? How does that work?
So, first of all, we only had one alleged parent to make a comparison to. And secondly, it's not feasible to develop a DNA profile from every individual. Then that might be 100.
Okay. So is this the strongest statistical number that is achievable in a DNA laboratory?
For maternity or paternity, yes.
So 99.9999%.
Yes, ma'am.
If I may have that second laboratory report, I'm going to need the Elmo, please. Madam Clerk, can we turn this? And up here, this report reflects your documentation of this case number?
Correct.
I'm showing Exhibit 1.70. Does that reflect the name of the individual whose sample was submitted?
Yes.
And that's what makes it a known sample?
Correct.
And then down here at the bottom, there is a listing of, quote, probability of maternity. Is that correct?
Yes.
And that's the reference you just gave us to the statistics.
Correct.
Thank you. Now, I would like to just ask you, you indicated earlier that there are some things that can impact DNA negatively.
Yes.
And I believe you said UV light, sunlight, some other factors. What happens with water? Can moisture in any way impact DNA?
Certainly, it's biological material, so it can be washed away.
And if I were to, my apologies, for example, brush against someone else, as I've just done in the courtroom, and I used my actual person as opposed to my clothing, can you describe for the jury what that is in DNA analysis?
We refer to that as contact or touch DNA, and there is a principle in forensics that there's a transfer of cells, however microscopic or small that amount may be, with each and every contact.
Is it possible that the contact leaves enough DNA to analyze?
Sometimes, yes.
Is it possible that it doesn't leave enough DNA?
Sometimes, yes.
And so initially in the processes you described, you said quantification?
That's fine.
Okay.
Yes.
My apologies to the jury. And amplification, correct?
Yes.
So if that touch did not leave enough DNA, would you know that?
Through our testing, yes.
Okay. And even if it left some, there still may not be enough to do anything with.
That's correct.
Okay. How do chemicals affect DNA, like cleaning fluids?
So different detergents and cleaning fluids are all going to have a detrimental effect on our ability to develop a profile from DNA testing.
What about friction, meaning rubbing or wiping? What impact might that have on DNA?
Again, it depends on the vigorousness of the friction or the washing, or if it's just a light brush or a very brief exposure. All those things are going to impact our ability to develop a profile.
In this case, did you do a run through the CODIS system of the profile you generated?
I was instructed to upload this profile, the profile from the 5 envelopes, from the human remains to the CODIS unidentified remains index. I did.
Did you have any feedback prior to receiving the known sample
I
from
did not.
CODIS? So nothing was in CODIS to compare her to?
Correct.
Okay. So ultimately, when you were able to get the known sample, is that when you were finally able to have confirmation of the remains?
That is correct.
I have nothing further, Your Honor.
Counsel cross-examination.
Good morning.
Good morning.
I'm Doug Weinstein. I represent Mr. Kassotis in this case. You were talking about contact DNA or touch DNA. If I pick up a bottle of Coca-Cola and I drink it, will I leave DNA around that bottle?
Theoretically, yes.
Where would that DNA be found on the bottle?
It could be found around the rim where your mouth has contact and where you grasp the bottle to drink it.
Would any possibly be found inside the bottle as well?
It's possible.
Okay. What about with a Coke can? Would you find DNA around that Coke can as well?
Again, similarly, it would depend, but around the mouth area where you take a drink and then where it's handled to pick up the can.
And you said that, I guess the elements like water or rain could in some way impact the finding of DNA, correct?
Yes.
But it's still possible to find DNA on a bottle or a can that has been exposed to the weather, correct?
Sometimes, yes.
And even if it's exposed to rain, correct?
Sometimes, yes.
Sunlight, correct?
Yes, sometimes.
Okay, thank you. I have no further questions.
Just, is it also sometimes no?
Correct.
So you don't know unless you actually have it, is what it comes down to, right?
And we perform the test. Yes.
Thank you.
Thank you, ma'am. You're excused.
Thank
I have 5 minutes to set up a computer.
Okay, and ladies and gentlemen, we'll let you step out while they set this up.
I will need to just.
Right.